happy that they are going home today? Raise your hand. So everyone blessed that they can be here only for a more time. Huh? Shall we conduct 2 more practical? Today, tomorrow and day after tomorrow? Huh? Okay. Anyway, so today we'll start with this session with a wonderful lecture that will be on nematode genomics. So the speaker, today's speaker is Dr. Vishal Singh Somanshi. He's a principal scientist, Division of Nematology, IRI, New Delhi. He is also PhD from IRI only, and he did post-doctoral research in USA and lot of experience in that genomics and related to etiology of the nematodes. So I'll just invite Dr. Vishal to conduct this session.
कुछ स्विच होगा ना मे बी देयर इज अ स्विच इन दिस हां स्विच होगा ये थैंक यू गुड मॉर्निंग एवरीवन I just asking how many people, like where are you guys from basically? I mean which universities, which states are you representing? All of how many states? 18 states. And all of your PhD and masters? How many masters? Please raise your hands. Who sent you here? No, I will say. Hello. Yeah. All right. I'm asking because these kind of trainings are very specialized trainings, and masters generally do not have an idea of what they're doing. And when it comes to life, PhDs have some idea about what they're doing in their lives. So these kind of trainings make more sense for the PhD students as compared to the masters, right? Am I right? I mean, the PhD students can tell me whether the master students are behaving properly or not. I just saw them coming in. So anyway, so today is the last day, and even if Dr. Diva has not told this, I know because they always keep my lecture on the last day. Really, the most boring lecture of the course. Let's put it in the end, and then we'll get done with it. इसके बाद और भी कोई है? I mean, you have any other lecture schedule after this? Dr. Ani Kumar. All right. All right. So I'm happy that you have reached up to this stage. It's a long training, staying away from your home in this humid and hot environment. Plus, Delhi has not been very kind this year. I mean, winters were extreme, summers were extreme, and then the rains you are seeing it firsthand. All right. I am Vishal. I am a scientist with the Division of Nematology, and today they have assigned me to tell you about nematode genomics. But how many of you had a nematology class before this? A lot of you know nematodes, man. Come on, this is 6-7 people is like a bonus. I was expecting only 1 or 2. So I'll try to take you through this. But my focus is going to be genomics. I mean, NIHEP has been very instrumental in promoting genomics in all disciplines of agricultural sciences. Now, nematology is a very small but a very unique discipline, and we are using genomics, transcriptomics to solve some of the questions that are central to nematological research. All right, but before I do that, how many of you know paintings? What do you know? Which artist made this painting? Okay, how many of you think it's a good painting or like it? Just be very liberal with raising your hands. If you will not, I will ask 3 times. You will waste a lot of time. I want to move forward quickly. Do you think it's a good painting? Yes, sir. All of you? Yes. All right. This is not a painting. This is the brain of mouse. कोई लगी ले दो मेरे पास. All right. So this is called brainbow. Rainbow. So what they have done is they have created a genetically engineered mouse. Okay, they inserted genes which code for different fluorescent proteins in front of all the neurons in the brain of the mouse. Okay, and then by some magical combination, okay, I don't want to go into that because that's a very complicated topic. When the neurons express, they express shades of, like you have in printer, no, 3 colors, but all the colors you can print using 3 colors, something like that, and then you can see all this. Now, why am I showing you this thing in the beginning? Because without genomics, this was not possible. And why do we need to see brain cells? Anybody can tell me why do we need to see cells? Anyone? Anyone raise your hand and just tell me. Otherwise, I'll start picking up people. Just wild guess. Don't worry about you are correct or not correct.
Like which part of the brain is getting activated at which
Exactly. Because nowadays people are mapping how your brains are responding to signals under a particular disease, condition. If you feel hunger, how your brain fires, okay? Or if you see some beautiful painting, how your brain fires. So something like that. But you have to know genomics if you want to know this, if you want to create something like this or understand something like this. Do you know what is this? What is it? Please be loud. Not a lot of people take nematode names, in this campus. So this is a C. elegans nematode. Why is it glowing green? Very good. But this was the first application. So he said GFP. So I'm assuming you know green fluorescent protein. All right. This is the first application, first transgenic animal expressing GFP tagged to the touch neurons. All right. There is a guy whose name is Martin Chaffee. Martin Chaffee created this worm by fusing a GFP construct to the nerves that are responding to touch sensation. Now again, if he had not had the knowledge of genomics or the genes of C. elegans, it was not possible. Later on, he went on to receive a Nobel Prize for this. Okay, along with, now why I would be showing you this picture here? Any guesses? It has a direct correlation to the previous picture. Bioluminescence, good, you are reaching there. Some people might have connected the dots already, but they are not speaking. Silence is not a virtue here, okay? Speak. No, C. elegans is not a marine organism. Huh? You have to be loud. Okay, close. Jellyfish naturally expresses GFP in the rinds. That's why the bioluminescence comes from. All right. So when Martin Chaffee was trying to create a reporter, so these are reporters. When Martin Chaffee was trying to create a reporter, he wanted a protein that fluoresces. Okay, and then he realized that there is a Japanese guy who is collecting bucket loads of jellyfish to isolate jellyfish protein that luminesces. Okay, so he contacted Shimomura. Shimomura gave him the sequence and the protein, and then the story evolved. 10 years down the line, they figured out that GFP is a barrel-like protein. If you excite it by blue and ultraviolet light, it absorbs it, emits green light. Okay. Now, it might sound very simple to you, but it is not. All right. Another guy, his name was Dr. Roger Chen. Okay, Japanese guy working in University of California, San Diego. He was a chemist. He is not a biologist. So Martin Chaffee is a molecular biologist working on nematode C. elegans. Shimomura has no idea what molecular biology is, but he likes green protein and jellyfish. And then Roger Chen, he is a chemist. He wants to tweak the protein molecules. So he created a rainbow by tweaking the structure of GFP. All these colors. So this is GFP, yellow, brown, orange, red, and then cyan, violet, purple. He created a range of fluorescent protein reporters. Okay, and together, so like since the discovery of it, now if you connect it all back to the first picture, These are the 3 people. All right. So if you connect it back to the first picture that I've shown you, their discoveries basically have led to the kind of reporting molecules that we use to visualize the gene expression inside a worm today. All right. All of which depended on knowledge of genes, proteins, and genomics. All right. So why is someone given a Nobel Prize? Any idea? Raise your hand and tell. You are all scientists. Nobel Prize is like Bahubali of science, right? So you should know what it is. Bahubali का तो सब scene याद होगा तुम लोगों को. Every scene you might remember, right? This is what tells me how serious you are about science. If I will show a random actress, maybe you guys will know, but I am telling you the name of. I am not even asking you who these people are. I am asking why is Nobel Prize important? What is the significance of a Nobel Prize? It is the biggest thing you can aspire to achieve in your life as a scientist. It is given to people who are alive. Number one, you don't have to die before you receive it. If your discovery is so significant that it changes the lives of thousands of people, and that's when you receive a Nobel Prize. Okay, it's not the money involved with this; it's the reputation and the kind of. Anyone know any other Nobel Prize winner related to your discipline? Please don't tell me you don't. Huh? So I can't hear you. You're murmuring. But yes or no? Raise your hands. Okay, what Nobel Prize winner do you know? What did she do? CRISPR-Cas9. Good. And she got it with some other person, right? All right. Any other Nobel Prize winner you know? RNA interference. You saying Andrew Fire and Craig Mellor? Oh, okay. Okay. All right. So I'm happy that you guys are aware, but try to be more aware. Okay. Do you know the month in which Nobel Prizes are announced? Pakka? No idea. December. Every year. Okay. And then they have a very nice. They announce things starting from October end. I think they literature का announce करते हैं, peace का announce करते हैं. Then they go on to do science, physics, chemistry, biology, and then they put up a very nice lecture. Like all of these people who are given this award, they go to a ceremony, and they all do a very nice lecture in which they explain in very basic terms to a layman how they won this award. It's a very nice thing to do. If you have time, please listen to it someday. You will be happy. Bringing you all to nematodes. Okay, this is how it looks like. Okay, a nematode is a very simple organism. किसी ने देखा है अभी तक? Any of you have seen it? You have seen nematode. So where did you see it? In a practical course. Okay. Anybody else? तुमने भी देखा है? Where? Okay, she has actually extracted nematodes from soil again, right? Okay, where are you from? AU. They have a department nematology there. Where are you from? MPUAT. They also have a nematology department, right? Yeah. All right. For all of you who have not seen nematode, this is how it looks like. So you are all caught up, and it's basically very simple structure. A tube within a tube. You can see it under microscope. Everything is visible. Nematode has no privacy. You can see how the things are happening inside nematode at all times. Like how is it eating, how is it pooping, and all that. But nematodes, if you go outside agricultural system, and I'm assuming all of you are mostly from agricultural universities, they are known as helminths, right? Or it's a general term meaning worm, and it encompasses a lot of things. So like for a lot of people ask me if earthworms are same thing or different thing. So earthworms are annelids, nematoda, they are different kingdom, they are phylums. Okay. Now this group includes flukes, cestodes, flukes, tapeworms, and roundworms. So helminths contain 3 different groups. tapeworm, roundworm and flukes. So flukes look like this. They are generally parasitic on the livers of animals. So if you go to any veterinary college, if you have a friend studying in veterinary college, they will tell you, yeah, we see them all the time, like they are parasitizing liver of animals. This is tapeworm. Now, a very common infection in pigs and all the animals that keep eating around in the garbage, sewage and all. But nematodes are a little bit different from that. Nematodes are cylindrical. So if you cut them from the middle of their body, like here, it's round. That's why it's called roundworm. Okay? And they have a very well developed digestive, reproductive, nervous and excretory system. So that's why they have been used as a model for so many things. Now, how many of you have seen Jurassic Park? Only one person, come on. It was a big movie in my generation. Your generation lost it. Now 3 or 4. है ना? हमारे टाइम में तो जुरासिक पार्क वाज लाइक द बाहुबली ऑफ दैट टाइम ना, डायनासोर पूरा इतना बड़ा बड़ा. आजकल आई सॉ ए वीडियो फ्रॉम आई थिंक हिमाचल प्रदेश और पंजाब. They have people wearing dinosaur costumes and dancing on the Punjabi songs. Like, you want to kill yourself if you see this. So the idea for that movie actually came from this guy. His name is George Poyner Jr. He's a nematologist. He works at University of Oregon. All right, and he likes to collect stones, ambers, and mummies. In one of the ambers that he collected, he found that there was a fly probably trapped in the gum of the plant. Okay, and in the gum, the fly was infected with nematodes, and the nematodes were trying to come out at that time. So nematodes also got trapped into the same thing, like this. So the fly is trapped, and you see these nematodes coming out of the fly. And it's a beautifully preserved sample. He's wrote a book. In this book, you see a lot of things like this, because nematodes are not, they are soft-bodied animals. They don't preserve well, so they don't fossilize. So we don't have a lot of history of nematodes, but this is where we find it. And the earliest note, so basically this is around 45 million years ago. All right, it's a lot of years, but you will be surprised. Nematodes have been around for approximately 600 million years. How many years have we been around on this planet? Any idea? कितने साल पहले हम लोग आए थे? You guys read books, right? It's a very simple question. As a biologist, if you don't know this, you have to go back today for 5 minutes and 2 मिनट का अपने आप के लिए साइलेंस रख लेना. क्यों? क्योंकि तुमने अभी तक, इतना सिंपल क्वेश्चन है. 3.5 million years ago we came? No, ma'am. Nematodes have been around for 600 million years ago. 5 million or billion? There is a difference of how many zeros? 3 zeros, right? So to my understanding, you're saying 5 billion years or million? 4.5 million. I am saying it right here. Nematode, this thing happened 45 million years ago. It's right there. So it has to predate this thing, na? We see the origin of vertebrates somewhere around 500 or 600 million years ago. I know it's a big number to wrap your head around, but everything has a perspective. Nematodes came around the same time, 600. Dinosaurs were around 300 to 250 million years ago. All right, not 200 million years ago. Human beings have arrived. If the history of life on our planet is of 1 day, we have arrived in the last 2 minutes. We are so recent. Okay, so that's why I'm saying sometimes it's really nice to go back and see how this history is not important because it's boring. We all hate history. Plant pathology ka history I hate very much, and nematology history is again very boring. But these kind of things put everything into perspective. Like, what are you dealing with here? So people who are trying to kill nematodes by using chemical pesticides, I tell them they are here for around 600 million years ago. You came back only like 15,000 years ago, and now you are trying to manage them. No, you cannot. Okay, But these are some samples. Again, that's why we should know history. And if you look at, this is the paper published in Nature a couple of years ago. How many of you what Nature is? It's a journal. Is there anything specific about this journal, or is just like your Journal of Indian Phytopathology or Journal of Pathology or something like that? Okay, he says high impact factor. Anything else? If you don't know this, please add 1 more minute to the silence that you will be keeping for yourself, okay, at the end of the lecture. How many of you know science? Cell? I have, so Nature, Science and Cell, right? If I have to publish my research somewhere, I will publish into those journals and then I can die peacefully, that I have done my job on this thing, right? Why? Because this is where, these are the journals that are highly impacted journals. Impact factor is what? How many of you published a paper? Research paper? तुमको तो पता होगा impact factor क्या होता है? What is it? Okay, value means if I pay 3,000 rupees for $3,000 for Nature paper, then any other explanation? If you have not published but you know the answer, you can speak. Your answer is oh God. Please add 2 more minutes. So the total minutes now are 5. Okay, go back to your homes whenever you have time. 5 minutes के लिए close your eyes and think what are you doing in science if you don't know these things. Impact factor of a journal tells you, okay, how many times the paper published in this journal this year have been cited in the previous year. Okay, so which means as a scientist, if Mendel said that these are the laws of genetics, right, and if I did my science and I did not find it happening like that, I will not cite this paper. So every time your paper is cited, that means some other scientist is referring to your paper and using your data or results on analysis. Okay, so the most highly cited papers are the ones that are used by everyone for their research. The Nobel Prize winning lectures are basically published in mostly Nature, Science and Cell. Okay, RNAi paper cited by more than 2,000 or 3,000 or maybe 20,000. I don't have the data yet. Okay, so 2,000 people thought that paper is important. Okay, I publish in small journals and no one cites my paper. Why? Because either my research is crappy or it does not make sense to a lot of audience. Okay, or my group is so small that only these people working in this particular group will cite my paper, right? And the higher the journal is, the more difficult the reviewing process is. Okay, so you cannot write. Like most of the people, what they do here is, you know, when the thesis submission time comes, all the results are compiled within the last 2 weeks. So you cannot write like that and submit it to Nature and think that it will be accepted, because as editors we know when the data is correct and when it's not. But these papers prove one point through multiple experiments, okay, and then they publish it here. So Nature has an impact of upwards of 35. I don't know what the current impact right now. Nature, Science, Cell, they have impact factor of more than 35. And please, I am not talking about NAS factors. NAS factor is something that we made up just to be happy, because most of the agricultural journals do not even have an impact factor. So we created impact factors to keep ourselves happy. NAS score of 5.6. 5.6 is nothing. It's below 0. Okay, so anyone who wants to do serious research, get serious about it. 5 minutes to please दे देना आज अपने आप को. Nature 2019, there was a paper. They basically surveyed the entire globe and they found out that, so you know these regions of world, I think everybody is familiar with the maps. The density of nematodes in the temperate regions is upwards of 19,000 per gram of dry soil. The number of nematodes per 100 grams of dry soil. And then you would assume that we are tropical countries, agriculture is happening, you probably will find more nematodes here, but it's exactly the opposite. You see all these hot and humid and tropical subtropical areas, they have less diversity of nematodes, 100, 675, 955 nematodes per gram of soil. The point I am trying to make it, they are everywhere. For every human being on the planet, there are 60 billion nematodes. All right, and there is another statistics, very interesting. It says that 1 in 3 person in this room is infected by parasitic worms, the animal parasitic nematodes. 1 in 3. Okay, so they in these areas the largest population of the world resides, and these are the areas where animal parasitic nematode infections are very common. Okay, so these nematodes come in very different shapes and sizes and forms. They are found everywhere. So this tells you that they are there on the Antarctica also. There have been expeditions. Some of my friends have been fortunate to go to Antarctica, and when they bring the permafrosted soil, the nematodes keep the soil thaws, and the nematode just start walking out of it, waking up after 45,000 years of sleep. This is amazing. 45,000 years. I think there was a in many popular newspapers there was an article about some worm, that Siberian worm that came out of 22,000 years sleep. So they are everywhere, and then they are found forest, oceans, wherever. In fact, so much so that if you remove everything else from this planet and leave just the nematodes, you will still see an outline of the major topographical features of the planet. All right, this is how they look like. This is an animal parasite, this is a C. elegans, this is a root knot nematode, plant parasite, free living nematode, animal parasite, animal parasite, animal parasite lodged inside a muscle cell, again an animal parasite, then this is a plant parasite sitting comfortably inside one cell of the plant. All right. There are more than 1 million nematodes. Expected diversity of nematodes is 1 million plus. Right now we know approximately 27 or 28,000 by name. All right, and of the ones we know, 50% are marine, 25% are free living, and only 25% are plant or animal parasitic. All right, but this is why we started studying nematodes in the first place. We didn't study nematodes because if someone is nice, we don't notice them. If someone is nasty, we notice, oh, this guy is nasty, right? So this is how we started getting into this. Now in humans, again, nematodes do not kill humans like malaria or AIDS or TB or these kind of diseases. So in the case of nematodes, the harm is measured in terms of million disability adjusted life years. So it's basically nematodes make you sick, so you start behaving disabled, you start being less productive, and they measure that thing, not the death part in this case. So it's measured in delis. And in plants, again there are many estimates. One of the estimate is like 157 billion US dollars per year. In India, there was a survey in 2020, and they put the figure in 10,200 crores per year to 30 Indian agricultural crops. All right, this is a very easy data to remember. 10,200 crores. Right? How many zeros are in there? How many crores you will get and you will be happy for the rest of your life? कभी सोचा? नहीं सोचा? You never thought about this? Oh my God! Half a minute of silence. Please add to the. If you don't know what makes you happy, right? Money makes me happy at least, and I have my favorite fantasies. If I find a bag full of money with no claimant, like it's like there, then what will I do with that? I can spend hours thinking about what I'm going to do with that money. So no, you guys are like either so evolved that you don't care about money or you don't know about money. All right. So the point I'm trying to make is 10,000 crore, 10,200 crore is a really huge amount of money. Most of us can live a very happy life in 1 crore rupees or 2 crores if you're living in Delhi. Okay. If you don't have expensive habits of, if you want to buy a Mercedes for 5 crore, then it's not. But an average Indian middle class family can live very happily within 2 crore rupees. Okay. Now, in case of animals, there are many nematodes that cause zoonotic diseases. Zoonotic means some nematodes can jump from animals to humans. Okay. Now, in case of plant parasite, I told you this data. We have lot of nematodes. If you want to go around in the rice fields here, you will find that some fields are patchy, and then most of those rice fields are infected by rice root knot nematodes, and then it causes approximately 2,346 crores of loss per year to the rice crop. It's becoming a major problem in rice nowadays. And then there was a survey somewhere some time back in the world. So what they did, they wrote letters to different people, nematologists in different countries, and they came up with this top 10 plant parasitic nematodes. How many of you know there is a journal called Molecular Plant Pathology? No? Half more minute to total minutes are 6 now. They do a survey for this, survey like this. So if you want to understand the top 10 bacterial pathogens of the world, I mean, you are entomologist, pathologist, there is no nematologist in there, no? So you are all entomologists or pathologists, or some biotechnologists also there. Biotechnology. So you will basically end up working with some of these things. Huh? So don't be, you're not special. You will come back to our systems. So whenever you want to say that this particular insect causes this much loss, how would you say it? You will write an introduction to your thesis, right? How will you say it? You will refer to Dhaliwal? No. That's a very old book. Or Agrios? No. So how will you say that these problems that you're working on are still relevant? Any one of you started working on your research? On any insect or fungus or pathogen? Just raise your hand. What are you working on?
Sir, I'm working on loose knot sarcoidosis in insects.
It's an insect.
Yes.
All right. Is it economically important insect or taxonomy?
Economically important.
Economically important. So how do you say it's economically important? Because for example, I mean, if you are talking, you can sit. If you're talking to some other scientist and trying to explain, right? And then you say, I'm working on this very cool insect called this, and it does this much damage. How do you tell?
So previous reports by different scientists.
So this is what I was trying to get at. Previous reports means generally this molecular plant pathology is a habit of doing the top 10 problems, like they do surveys. And in case of nematodes, they did a survey in 2010, and then they found out root knot nematodes, cyst nematodes. Root knot nematodes are Melidogyne, cyst nematodes are Heterodera and Globodera, root lesion are Petilencus, burrowing nematodes are Rhadophilus, Ditelenchus dipseci, pine wilt nematode Bursaphilencus, reniform nematode Ziphinema, Nacobus, Aphylincoids. So like top 10 nematode problems of the world. All right. Now what happens here is, you also have to look at this. This is the trinity of animal parasitic nematodes. They are basically, these three nematodes are responsible for more than 90% infections of human beings. This is 2000, year 2000, 20-year-old data. Okay. Ascaris lumbricoids, I think most of us who are growing in small towns or villages have seen this nematode. This is the worm for which you take a 6-monthly deworming tablet. This one. Okay. Second one is whipworm, and third one is hookworm. All right. So again, 2.2 billion people. This data is old. Right now, this data might be alarming. Ascaris, whipworm, and now the beauty of this data is, and then we are coming to genomics in 2 slides. This number surpasses the population of Europe and Russia, North America, number of new cases of cancer per year, and diabetes or malaria. All right, this is such a big problem, but because they do not kill, they are never a priority. Okay, priority for research funding, research projects, and all that stuff. Everything is linked to money. So if you thought I was trying to be funny when I was talking about money, no. The quality of your research is directly proportional to the quality of money your boss will have or your lab will have. If I am wrong, all right. So if I get a bag full of money, I also think about what I am going to do in my lab. No, it's not like I am going to buy a yacht and go sailing the world. So again, everything is related to money. But most of the nematodes, as I told you, 50, if you remember that pie chart that I have shown in the beginning, most of them are menine. In soil also, majority of nematodes are good, and they do what? They do nutrient mineralization, they they keep moving the soil, churning the soil, they regulate the population of plant parasitic nematodes, they are used as bioagents for managing insect pest of crops, and then our model organism, the Hrithik Roshan of all the nematodes, Caenorhabditis elegans. Okay? It has been the poster boy of nematology research. Till now, approximately, not approximately, 4 Nobel Prizes have been awarded to people who are working with Caenorhabditis elegans in one way or other. Right? Now, this brings all of it, right? This introduction. Abhi to finish ho raha hai introduction, and we have 20 more minutes to go. Now, it will be easy for you. The central problems in nematology research, as per me, it's like personal thing. If Dr. Diba was a nematologist, she might have some other thoughts about it, and this is a very personal thing to me. I think that there are too few chemicals for management of parasitic nematodes. Why? If you talk about drugs, can anyone tell me how many antibiotics are available in the market for human diseases, human bacterial infections? How many? I am talking, not asking you the name. I am saying how many? I am saying hundreds. होंगे 100 से ऊपर, more than 100. In case of parasitic nematodes, you only eat, if I will ask you one tablet that you have been eating since you were a child, it was albendazole. Right? Even now, if there is a deworming program, they give you albendazole. Okay? So, one drug and 2 or 3 more, one of which became popular in times of corona. You remember ivermectin? Everybody is eating ivermectin. It was not a disease for virus, a tablet for virus. It was a tablet for worm, parasites. But so one of my problems is there are too few chemicals for management of nematode. Even in case of plant parasitic nematodes, we have only, I think, 4 nematicides that have been registered for use as a nematicide as of now in India. Only 4. Insects के लिए कितने हैं? How many for insects? Many? Yeah, more than 20, right? For nematodes only 4, and of these 4 also, you cannot use many of them because they don't have a label claim for many crops. All right. So this is one problem. Second, again, my understanding, I think nematodes are very diverse and complicated, but they are very poorly understood. Like, won't it be amazing to understand how a nematode can be in a sleep for 25,000 years, right? And it comes out absolutely fine. Then we don't know what is the contribution of free living nematodes to environment, and then how would climate change affect the plant nematode diseases. All right. So these are some things that I think are important. Again, it's a very personal thing. Science is a very personal thing. You view problems as per your own this thing. This brings me to genomics, because we have been using genomics to answer some of these questions. All right. And then by now you must be very clear genomics. So I am not going to go into fundamentals of genomics. My lecture is overview of nematode genomics. All right. And of that 40 minutes I have already spent in telling you what are nematodes. 20 minutes I will tell you what is the genomics happening in this thing. So in my opinion, genomics is again structural, comparative and functional. There are 3 subclasses or subdivisions. But whatever we do, it must be useful, it must be for some purpose, and it must help in answering a biological question, like knowing your future, DNA screening. All right, this is reality. I mean, nowadays in human medicine, things have come down to sequencing your genome, understanding what genes are wrong or right in your body, and trying to correct that. In case of structural information, what we understand basically is we use that information, genomics information, for species diagnosis, identification, to understand the population diversity, and to recreate phylogenetic relationships. Okay. And then in case of functional information, we use genomics for understanding gene functions, host parasite interaction or pathogenesis, gene evolution, gene polymorphism, and it's basically being used from the diagnostics to creating brain bows. Okay. I think brain bow is one of the most complex applications of genetic manipulation I've ever seen. One of the most beautiful also, but it has simple uses also. For example, most of you work with different pathogens and you probably use some molecular marker to identify what that particular pathogen is. Some of you, for example, in case of virology, there are specific kits available for diagnostics of your viruses. All that information is basically derived from genomics. Okay, if you want to create a kit, you have to know the genomics. You have to know the genome of your pathogen or insect. Okay. Now, so I mean, I am sure that a lot of people have covered these answers, but how it is helping us in nematology research? So we are studying genomes, and then it helps you in answering so many unique questions. For example, humans have 99% identical genomes, but we look so different from each other, and right, we don't know why, because again genomics research has just started 1998 onwards, right. C. elegans was the first eukaryotic organism to be sequenced as a pre-run for human genome. All right. So it started in 1998, and we are not even 25 years into genomics research, and we have understood so much, but whatever we even know now is just the tip of the iceberg. I mean, there is so much going on in the genomes, we don't have any clue. Again, we have different species and races of pathogens. They differ in virulence, host range, climate adaptations. Why? I don't know. Probably the answer is somewhere in the genome. People have been trying to attempt solving genomes, understanding genomes of their favorite pathogens or organisms. So maybe we'll have some answers. So this is the 11 December 1998 on the cover of Science, C. elegans, sequence to biology. All right. How many of you know what is central dogma in biology? DNA to RNA, RNA to protein. All right. We still at 6 minutes, huh? So otherwise this would have added another 3 minutes, come se come. All right. So I told you that this was the guy, Dr. John Sulston, who was basically instrumental, and they, again, you have to understand, they did it as a pre-run for human genome sequence. Okay. So he is the person who was responsible for it. He was the director of founding director of Sanger Center. Now, okay, the last 15-20 minutes maybe we are talking about the actual part. So I told you about 27,000 nematodes we know by name. Out of that, 10 are very, very important for agriculture. Maybe 5 or 10 are important for animal diseases or human diseases. As of now, sequence of 164 nematode genomes has been sequenced. It covers important animal parasites, plant parasites, free living nematodes. Then there is another project going on to sequence 959 nematode genomes. Then there are many nematode genomic databases from which you can obtain parasite specific, or you will live na. Okay, if you want to go out and just walk it off. All right. So for example, we have a very useful resource called WormBase, Worm Atlas, Worm Book, Worm Wiring. So Worm Book is a collection of literature freely available to anyone who wants to access it. They do not even want you to do an account sign up. You can just go there, download it. Worm Atlas, again a free resource to understand any structure inside the worm. Then you have some databases here, but mostly what we use is this one. Where did it go? This one. All right, it's called WormBase. So in United States and outside India, people call them, affectionately call C. elegans as worm. So everything is a worm, like WormBase, Worm Lab, Worm Atlas, Worm Tracker, whatever. All right. Now in India, worms means kida, and kida can be anything. It can be an insect, it can be earthworm, it can be nematode, or anything that moves. So here we have 163 nematode genomes belonging to 130 different species, and 38 institutes have as of now they are working on this kind of this. 38 institutes have contributed to this. Now in India, we have been involved in doing a lot of genome sequencing and transcriptome sequencing work, and again it's for useful purposes. I will give you one example to show what we do. We have sequenced 5 whole genomes, we have sequenced 14 nematode transcriptomes, and then we are using a lot of that information to come up with new tools for managing nematode diseases of crops. We have done rice root knot nematode Melanogain graminicola, cereal cyst nematode Heterodera aveni, entomopathogenic nematode Heterodera indica. Then we have also done one insect Maruca vitrata, and then because we also work with rice and nematode interactions, so sometimes you have to, you will have to understand this thing, that as you grow up in science, you will not be confined to one particular area. So I am not a plant biologist by training, but I am sequencing rice just to understand why some varieties are resistant to nematodes and why some varieties are susceptible to nematodes. All right, so we have to do this kind of crossovers, and this is all what we have done. A lot of information here. Now I will give one case study about how we apply this knowledge actually. So I told you Melanogain graminicola, so they are managed by using toxic chemicals. Then for example, the most commonly used chemical, so one chemical that we still recommend to everyone is carbofuran, right? What is carbofuran? What is carbofuran? It's a general biocide, it's a poison. If you eat it, you will die. So we say for insects, they say 4 to 5 kg AI per hectare. For nematodes, we say 1 to 2 kg AI per hectare. But if you broadcast it in such a high concentration, snakes, frogs, lizards, birds, everything dies. Why? Because it's very toxic, right? And because of that toxicity, these chemicals are being banned or being phased out till we find some better alternative. Now again, I told you that there are 4 chemicals that have been registered by some companies in recent years, last 2-3 years. Unfortunately, none of them is a true nematicide. So one was a fungicide sold by Bayer Crop Sciences with the name of Velamprine. It was a fungicide for which they found a serendipitous nematicidal activity, okay, and it was easy for them, so they launched it as a nematicide in the market. But the problem is it's phytotoxic. Phytotoxic means if you spray in more than recommended dosage, it will kill your plants, and it's very expensive. Carbofuran was cheap at least, okay. So now how nematodes find their way in to the host is basically they have lot of receptors on their body. They sense where the roots are, they sense the exudates, and then they move towards those roots and infect those roots, okay. So what a smart approach would be to stop them from finding their host plants in soil. So we can, like, you can put an odomos on you and then mosquitoes won't be able to find you, right? How does that work? Because mosquitoes track you by the carbon dioxide or the specific body odors that we emit, and they are trained to find us by that. So if you apply a odomos, then you will be fine, okay, or any other thing that changes the odors of your skin. Now this is how root knots look like. If you can see, this is a tomato plant and lots of galls in this area, okay. So how we select genes is basically, here I am showing you what is the life cycle of nematode looks like, okay. How nematodes infect. This is a nematode worm, juveniles, okay, in the soil. They will get attracted towards the roots of the plants. They will invade. After invading, they will start developing their feeding sites and make galls like this, which will basically allow the nematodes to reproduce, and then the females will lay eggs, the juveniles will again hatch out from the eggs, and this is how they will keep infecting in the soil, right. Now there are some resistant stages in here also. For example, these juveniles are very hardy, they don't generally die easily, and these eggs, they can lay in the soil for a very long time. But now it gives you a sense of what you can target if you want to manage nematodes. All right. For example, what if I stop these juveniles from finding the roots? Okay. What if I stop nematode feeding cell development? Okay. And then what if I somehow manage to induce some kind of lethality when they are laying eggs? So it gives you a sense of where you can target. And then in our previously in our group, we found that there are neuropeptides that help nematodes find their hosts. Neuropeptide means some chemicals that help in the signal transmission in the nematodes to perceive their environment. And then if you want to block nematodes, then what do you have to know? How many neuropeptides are there in a particular nematode? Okay, because nematodes generally have copies of genes, they don't rely on one copy of one gene. So even if you end up blocking one neuropeptide, okay, they have 5 more, so they will keep working. So you have to know how many neuropeptides are there, you have to know what is the sequence of that neuropeptide, then you have to understand the expression pattern of neuropeptide during different time points of infection, and then only you can even think about coming up with a credible strategy to manage nematodes. So in addition to these kind of strategies, you can identify metabolic choke points. Choke points means there are pathways going on inside any pathogen, it can be your fungus, bacteria or anything, and then you will find that there are certain genes which are bottlenecks for any pathway. Bottlenecks means a lot of things are happening through that particular gene or enzyme. So if you identify that and target that and block it somehow, you will be able to disable or debilitate the nematode pathogen or pathogen whatever you are working on, because it will stop downstream processes from happening. So these kind of things are called metabolic choke points. Now how will you learn choke points? So for example, if I sequence one nematode or two nematode, that's good, but all the people who are doing 959 nematode genomes, right, what are they going to do with this information? They will lay all these nematodes pathways side by side, they will figure out what are the pathways that are specifically operating inside the nematodes, and they will target only those. Extremely safe methods for management of nematodes without harming any other organism in the soil. All right. So in our case, we ended up silencing about 30 genes in incognita, 40 genes in Heterodera aveni, 45 genes in Graminicola. In Maruca also they have worked on approximately 10 genes, and then they found that silencing all these neuropeptide genes basically ends up giving you certain kind of like 60 to 70, 80% kind of control in the nematodes ability to find host plants. All right. So when you disrupt these genes, nematodes are unable to find their hosts. Then genomic information also helps you in identifying different targets. Targets means all of these are, so neuropeptides is just one class of targets. All right. If you look in your target genes, your organisms, you will find that there are many other genes, for example, small ribosomal proteins, cotomers, pre-mRNA splicing factors, spliceosomal SR proteins. So basically there is a list of more than 50 genes that we already know that can be used as targets for managing plant parasitic nematodes. If you hit those targets, now if you are a chemist, you understand what the protein structure of that particular gene, come up with a drug molecule that can inhibit that particular protein and spares everything else. Okay. So this kind of information, they are targets. Once you know what your targets are, you can design RNAi based management strategy, genome editing based management strategy, chemicals based management strategy, or you can do n number of things. But all this information basically comes from knowing genomics. Now पक्का ठीक है. So I'll just one case study I will tell you and then we'll stop. Okay, I know it's going on for more than 1 hour. So this is rice root knot nematode. Again, our lab works with this. A healthy field will look like that. An infected field will probably look like this. If you go around just outside in these rice fields, you will find a lot of fields which are looking like this right now. Okay, and if you uproot these plants, the healthy normal roots look like that, but the diseased roots will look like this. They have these are galls, and then rice root knot nematode is known to produce terminal galls. At the end of the tip of the root there will be a gall, swelling and gall. Okay. Now this is seedling in which you will see small galls, very fine, very small galls all over the place, there, here, and in the big ones at maturity you will find big galls, this. So like the galls sometimes become like very huge and swollen. Yeah, so we started working on this particular nematode from 2018 and we did a genome sequencing and we figured out that the genome size was 38.18 MB. It contains around 10,196 proteins, but we improved the sequence again in 21. The genome size got smaller and then we found that it's actually coding for 14,062 protein coding genes. Now why do we have to do this? Because again, you know, with passage of time technologies improve, and then if you combine lot of technologies platforms together you get better results. The better results means you will have an accurate idea of what is going on in the genome, and then your targets will be identified more accurately and your strategies will work more accurately. If you don't know the complete genomes, so how many of you know the difference between complete genome and a partial genome? Maybe we'll add the 3 minutes here now. ये जीनोमिक्स का नायब है ना, इतना तो अभी तक तो पढ़ा, किसी ने तो बताया होगा. Okay, we'll come back to this question at the end. I don't want to take more than 5 minutes now. So again, we got a genome completeness of around 88.6%, and then we identified a lot of targets in this genome. So like for example, we identified a lot of effectors, we identified 561 secreted proteins of Mylidocaine graminicula that we can target, that this nematode uses to cause infection in plants. Then we identified, there are approximately 66 previously identified effector molecules, out of which 35 we could identify in our nematode genome, and then we identified approximately, you know, all these numbers, 1853 proteins that might be interacting with plants to cause disease. So all this is basically getting us more and more targets to follow upon. Then nematodes degrade carbohydrates, they have lot of carbohydrate degrading enzymes which they have acquired from lot of bacterial and fungal pathogens. And then we found that Graminicula has approximately 1575 carbohydrate degrading enzymes, that because they move inside the plant root, so they know they require all those to dissolve the cell walls to create a feeding site and all that. And out of that, 5 of them were unique to Graminicula. So maybe this is what is giving it specificity in terms of infecting rice as opposed to other different crops. And then we identified the known targets, and then in summary, I mean I have told you the summary only, so don't have to go back to it again. Now what we are doing right now is, all these identified proteins represent putative targets that may be utilized for development of novel chemical molecules and approaches for management of Graminicula. All right. Now in some of the cases, we basically identified one target which was basically a fatty acid metabolism regulator. All right, we silenced it and we wanted to see if we hit that target, is it gonna affect the nematode biology and development also. Okay, then we took the RNAi approach, and this is the transcriptomics. So as soon as you silence that particular nematode, because you have to test your hypothesis by also understanding whether the target you have hit is working in the way you expect it or not. And how do you expect that? If we are hitting a fatty acid regulator, it should affect the fatty acid pathways. Okay. Here we see that as soon as we hit the target, a lot of genes have turned red. All of these fatty acid metabolism, fat metabolism genes, they stopped functioning as soon as we hit the target. All right. So this is the information that we generated using a transcriptome experiment. Okay, this is not a genome, this is a transcriptome experiment, and we found that yes, when we hit this particular regulator, the fatty acid metabolism is getting affected, and then we found that it's also giving us significantly less infection of roots, significantly altered biology of the nematodes, less number of eggs, smaller females, because fat reserves are gone. And then we proved it in root knot nematodes, but this approach, because now it is a target that we have validated, this approach can be used for any other nematode pathogen that you want to target. All right. Now, last two slides, and one of them is thanks. All right. So, I want to really all of you to pay attention to it. I know after 1 hour of continuous speaking, it is very difficult, but take a few deep breaths. Last 3 points. Okay. Number 1, I don't know what have you learned before that, but personally what I feel is it is a very powerful tool to investigate a range of biological questions and life on our planet as we know it. Right? There is no end. A lot of people, brush away molecular biology, genomics as, oh, they have money, they are doing fancy stuff. I want you to get above that now. Just give a thought to why these people are doing it. Yeah, because when we are students, we tend to become cynical and we tend to become negative more than we are positive. Why? Because if his boss has a molecular biology lab and my boss has not, then I start thinking that I am at a disadvantage somehow. Right? All you have to do is go to his boss and tell that, boss, I also want to use your molecular tools for my research. In nematology, we have different, like 4 major areas. One is taxonomy, another is entomopathogenic nematology, one is management, another one is molecular nematology. Right? Somehow they have big boards if you visit my division. And all the people who are assigned the rooms in molecular, nematology, there used to be a lot of happiness among the students. Now I got the boss who is working in molecular nematology. This thing. Okay, so what? I mean, I was working in taxonomy. She works on taxonomy. She uses molecular biology more than any other person I know. Right? So it's not the question of, these are all tools. Even for taxonomy, you can do genomics. All right. These are all tools that you have to use for your research to find some answers. All right. So there is no end to how much you can use it in your life. And then it's being used to develop better methods for pest diagnosis and safer methods for management of agricultural pests. We are using a lot of genomics information. She is also using it for development of new diagnostic methods. Right? All of you remember na corona ke time mein how many kits were there. So the most prominent kit was RT-PCR, and then later on in the 2 years that disease pandemic was there, people came up with CRISPR-Cas based kits and lateral flow dipstick kits and so many kits. Right? Now there itself you have 9 ideas on how to develop kits for your diagnosis of your pathogens. Right? In agriculture we have this problem, because if someone wants to identify a nematode and they want to be confirmed about it, they send it to me from anywhere in the India. If someone wants to do a fungal identification, they are sending it to her. So half of the time we are spending in what? Trying to do samples for other people for free, because if you ask them for money, they will say, we don't have money, we are students. I know, that's why I want students to think about money. Okay, most of the students do not know that they can apply for different fellowships and support their PhD research, and maybe maintain a car also in parallel, because you never thought about that in that way. In United States, everybody who gets into a PhD gets a fellowship, they maintain their car. It's a lifestyle for them. PhD is not like a poor student thing for them. PhD means they have a life. They are working in a lab, they have a life. They have a car, they live in an apartment, and they also work. and they are much better scientists than 80% of the scientists working in this institute. I am not talking about agriculture universities. वहाँ तो 99.9 हो जाएगा. PhD students, just like you, coming into the PhD programs, first year when I see them and the 5th year when I see them, they will be better than 99% scientists of our entire NAR system. It's not a small claim I am making in front of everyone, and they are probably putting it on YouTube also. All right, it's because it's all about the attitude. In India, we always think, I don't know, half of the people don't know why we are doing PhDs. Right? They have no idea, no clarity. For example, I asked you certain questions. You still owe yourself a 6-minute silence, right, after the end of this meeting. But if I start asking why, a lot of you will be lost. So there is always a goal to things. When you have a clear-cut goal, you prepare for it and you achieve it. If you do not have a goal, you are lost. All right. And then again, third point is basically a summary of these two only. It may be used in a variety of ways to achieve different end results. It's not that people who are doing genomics can use it only for their research. I mean, if they have created a resource, anybody can use it. All the 164 nematode genomes are available for free. Whatever genomes they have sequenced for fungal pathogens, it's available for free for everyone to use. Right? All you have to do is elevate your game, think about how you can use it, just use it. And we did not sequence C. elegans genome, but we use it every day. Right? So this is how it works. And with this, I first of all thank you all, because the last day I know it's tough living here in a different place for so many days. And then I acknowledge the funding from some of our funders, and to NAHEP also, because they have been very kind in supporting my students. Thank you all. If you have any questions, we will be happy to take it. 6 minutes, ठीक है? No, it's not a joke. I was serious about it. Whenever we say we are a scientist, okay, I don't know about you, if you are not being true to, so for example, I mean, we all know Manu Bhakar now, right? We all know Nikhat Jareen now. They have achieved something that has made everyone proud. All right. So my question to you is this: they have not become what they have become by being lost about what they wanted to do in life. They knew that they want to do this in their life, and they have spent entire year. So from the time you decide you want to do a PhD, your objective should be very clear. And if science is a game, knowing what impact factor is, knowing what journals are, knowing what funding is, knowing how to use grants, monies, like this is the game of science. So you have to understand it. If you are not doing it this way, you are basically preparing to fail. And then you will, 5 years down the line, you will say, we don't get jobs. Why will you get a job? Half of the people don't know the basic answers of these questions. Right? You are very fortunate in India that we still have ARS examination that makes you a scientist. In no other country in the world, there is examination that makes people scientists. They look at what you have done as a PhD student, then only they make you a scientist. So this is that serious. You are fortunate enough that lot of money is coming into agricultural research. NAHEP is one of those projects that worked because they got a lot of funding, and they got a funding because all the people who are thinking about getting this kind of funding into agriculture. All right, we are all beneficiaries of that. We did not create that project or got that money. So someone here in my institute, in your university, they worked to create a project like that. They went to the government and they said that let's bring some money to train all these students from different parts of country in genomics. That's why you are here. Even for 50, how many? 21 days?
10 days.
10 days. Not even your Mossy will keep you in your home for free.
Well, Vishal, there are around 600 applications.
So you understand the enormity of this exercise. They might have spent upwards of 10 lakh rupees in this in 10 days. 15 lakh rupees. Right? So somebody thought about money. So please think about money if you are not thinking about money. That's the point. Thank you very much. If you have any other questions, you can speak now. Or if you have any other question about nematode genomics, you can just email me. Okay, if you just remember my name and find it on IRA, you will get my email ID. All right.
A simple exercise they should do. Other day is that the manual also for the
Yeah, yeah.
everybody.
It's all right. There is only one nematologist in the country by my name. So even if you Google it, you will find it. All right. Thank you so much. I don't think that, they're asking anything.